Protein synthesis, the process by which our cells make proteins and why it is essential for life. To start off, we have DNA. So DNA is made up of nucleotides, each containing a sugar, a phosphate, and then a base. The nucleotides link to form a backbone, and two strands twist into a double helix held by hydrogen bonds. The base pairings would be adenine with thymine and then cytosine with guanine. Then we have central dogma. Central dogma is a process where DNA gets copied into RNA, to which that RNA is used to make proteins. This process is what makes sure that the genetic code stored in DNA actually turns into working proteins, which is super important. So in transcription, it takes place inside the nucleus, and then DNA is used as a template to make messenger RNA, also known as mRNA. Some of the key components would be DNA, like we just talked about, and then ribonucleotides, as well as RNA polymerase. Some steps of the transcription would be initiation, and then elongation and termination. So to start off, during initiation, the DNA unwinds. RNA polymerase attaches to the promoter, separating the strand so one can be read, which is what's happening right here. And then in elongation, RNA polymerase moves along the template strand, matching RNA bases with DNA bases. And the RNA chain gets longer, which is what goes on here. And then in termination, the polymerase reaches the end of the gene. The DNA rewinds back into a double helix, and then the pre-mRNA releases, which is what happened here. So in translation, we have some key structures would be ribosomes, tRNA, mRNA, and then the amino acids. So then in translation, we begin with initiation, where the ribosomes, mRNA, and tRNA assemble. The small subunit finds the start codon, AUG, and this large subunit joins, placing it in the P site. So then during elongation, tRNA brings amino acids to the A site, where peptide bonds form and the ribosomes shift as the chain grows. And then finally, in termination, a stop codon is reached and a release factor binds, and the ribosomes separate and the completed protein is released, which is what happens in this picture right here. Like we said, some of the key components, mRNA, tRNA, ribosomes, and amino acids. The mRNA carries the genetic message from the DNA to the ribosomes, so the cell knows what proteins to make, which is what's happening here. The tRNA, it acts like a helper, bringing the correct amino acids to the ribosomes during protein building. The ribosomes, they are the place where the instructions are read and the protein is put together, which is super important. And then the amino acids are the small building blocks that link together to form the final protein. So like I said earlier, protein synthesis is very important because sometimes when proteins aren't folded the right way, it can cause illnesses or diseases. One good example I chose for my disease would be Alzheimer's. What happens in Alzheimer's is the protein called amyloid beta, it doesn't fold correctly, and the ends end up clumping together in plaques in the brain. So these plaques block the brain from talking to each other, which leads to the memory loss and the other problems and symptoms that come with Alzheimer's. Alzheimer's disease is one of the illnesses that just shows how important protein synthesis is for our bodies and how it is important that it works correctly, or because if it doesn't, it can cause diseases or illnesses.